Good afternoon, everyone. My name is Manoj Dadlani. I'm the CEO of Cosmos ID. I think we'll just give everyone a few minutes to log in and then we'll get started. Okay, well, let's get started. So thank you for joining us today. Again, my name is Manoj Dadlani. I'm the CEO of Cosmos ID, and welcome to our seventh webinar in our webinar series focused on the microbiome. Delighted to have Dr. Dele Ogunremi with us, and he's going to give us an amazing presentation on some of the recent work he's been doing. Just before that, I'll give you just a quick overview on Cosmos ID, and then I'll hand it over to Dele. So Cosmos ID has been in the microbiome business since 2008. We've been focusing on reducing the bottlenecks in metagenomics in various ways, and a lot of it has to do with increasing the power of studies and reducing bias through the entire process of metagenomics and microbiome sequencing and characterization. And that process, again, is end-to-end, so it includes study design, sample collection, preparing samples, regulatory compliance, bioinformatics, both taxonomic and functional bioinformatics, and then interpretation and support in publications and translation. And some of the core technology that we've built at Cosmos ID has really been around the bioinformatics side. We are the world's leader in accurate strain level resolution. We just actually had a database update, which brings us to 170,000 microbial genomes and human sequences. It's the largest curated database in the world. We've developed proprietary algorithms that allow us to have very accurate sensitivity and specificity. And then on top of that, we've layered a lot of machine learning onto our algorithms to improve that accuracy. And when you're looking at shock and metagenomics, you're able to analyze bacteria, viruses, phages, fungi, protists, as well as antimicrobial resistance genes, virulence factors, and functional genes. We have customers all around the world and many peer-reviewed publications. So here's some internal benchmarking data where you can see the accuracy of Cosmos ID's analysis for some of the common bioinformatics pipelines in the metagenomic space. And what you can see is at the strain level, you can really see this big differentiation between precision and recall, or sensitivity and specificity of the Cosmos ID algorithms and databases versus some of the other common tools in the space. So it's easy for us to say that our bioinformatics is really strong. We've also had some amazing third-party validation. We've won both of the Precision FDA challenges in strain-level identification, as well as Janssen Pharmaceuticals Mosaic Strain Challenge. So we also have some great third-party results to show that accuracy is best in class. We've made it really easy to use, so we have a simple cloud-based platform to simplify the analysis and comparison of samples to each other. In addition to metagenomic samples, we also do isolate strain subtyping, looking at whole genome sequencing of isolates and being able to compare similarity and differences of isolates, and also look at transmission events. And then finally, as a service, we offer sequencing, we offer bioinformatics software, we also offer custom bioinformatics support. So if you're looking for kind of that extra in-depth analysis, it's something that we can support as well. And here's just some examples of our amazing customers and partners that we work with in the pharmaceutical, nutrition, clinical, animal health, cosmetics, supply testing, and research industries, as well as our government partners. So again, thank you for all of your great support. We look forward to working with you into the future. And now let's kick off this webinar. So the title of the webinar is Development of Universal Detection Methodology for Foodborne Microbial Pathogens. And Dr. Dele Ogunremi is going to honor us with the presentation. He's a research scientist at the Canadian Food Inspection Agency. He spent 25 years developing research programs and projects in animal health, food safety, and genomics. A huge list of accomplishments. He's developed over 30 diagnostic protocols, 50 research projects, 50 innovations, a DNA vaccine for avian influenza in chickens, and 2 patents and 1 more patent pending. And in addition to that, he's an adjunct professor in the Department of Food Science at the University of Guelph in Canada. So with that, I'm going to hand it over to Dele, and hopefully this echo will calm down too.
Well, thank you very much, Manoj. Am I coming through?
Yes, you are.
Great. And is my screen showing? Do you have my screen on? Can you see me?
You need to switch to your presentation.
Okay. All right. Am I visible now? Can you see me?
Perfect.
Excellent. Well, thank you for that kind introduction. It's a pleasure to be here today to talk about our collaborative work developing a universal detection methodology for foodborne microbial pathogens. And by way of introduction, I intend to go through the importance of food safety. I realize most of my audience probably know quite a bit about food safety, but I don't want to assume everybody is on par, but it's important to put the talk into that context. And then I'll dwell on the application of genomics and bioinformatics to food safety. And the focus of my discussion today, my presentation, is a culture-independent diagnostic test. And I'll go through ways in which they are approaching developing the test for bacteria, viruses, and parasites, using spike lettuce, and then how we verified the performance of the test as a prelude to a full validation, which we are still undergoing at the moment. So I won't be sharing our validation data yet. And then I will show some of our results from a simulated field testing. 1 in 8 Canadians are known to suffer annually from foodborne illnesses, according to the Public Health Agency of Canada, which is the counterpart for the CDC. And we know a number of common pathogens that are responsible for this situation, and they are shown here. However, there are a lot more. At the same time, if you go globally, we see that foodborne illnesses is responsible for as many as about 500,000 deaths every year. That is the size of the 10th biggest Canadian city, that's Kitchener-Waterloo. So that's wiped up the surface of the earth every year because of foodborne illnesses. And I actually would like to think that's probably an underestimation, because we know that there's an estimate for just one organism, Salmonella, non-typhoidal Salmonella, that's responsible for as many as 155,000 deaths every year. Yes, we do know that we're dealing with an issue that results in hospitalization and in deaths. But there's also economic aspects to it. This is a fairly old slide, but it hasn't changed by much. what you're seeing there is the cost of product recalls, the most expensive recalls ever. By the time we get the data from Boeing 787, maybe that will go on top of the list. But again, that won't change things by a whole lot. But what you see there is foodborne illnesses, or at least one foodborne illness, has been responsible for one of the costliest food recall ever, costing about $1.5 billion. And I must also add that the CEO of that company is currently in jail because of what happened there. So there's an enormous cost to foodborne illnesses. We're dealing with viral pandemic right now, but foodborne illnesses can also cause such a devastation that at least in one case, that's about 20 years ago, there was a concern that the spread of Salmonella enteritidis may be approaching a pandemic. And that situation, even though has abated somewhat, Salmonella enteritidis is still one of the most prominent causes of foodborne illnesses. Now, this slide shows 30 common pathogens that have been identified in Canada through hospital records to be responsible for foodborne illnesses. And the bacterial agent tends to cause the most severe hospitalization and death scenarios. But if you look at that same publication, we're told that there are many more pathogens responsible for foodborne illnesses than the 30 that are commonly tested for. And these are identified as unspecified agents. So these are not tested for, they're under the radar, but they're causing more illnesses. So that tells me that we'll need a different approach if we really want to overcome the challenges of foodborne illnesses. Canada has one of the best foodborne illnesses, food safety records in the world, yet we still suffer from as many as 4 million foodborne illnesses every year. So is it possible then to look at a situation where we can begin to address this issue in a very substantial way? Or is this a dream? Is this a Sisyphean challenge that may not be corrected anytime soon, or could even just be frustrating if we even attempt to address it? Or can Sisyphus get a Hercules to help out? Because this is a Herculean task. And I would try to suggest that microbial genomics or genomic sequences are Hercules when it comes to addressing the challenge that we have with foodborne illnesses. Now, there are a number of issues associated with food microbiology, with detecting microbes in food, in contrast to, let's say, clinical symptoms, clinical samples from humans that tend to have abundance of microorganisms. Some of them are listed here, and I won't go through them in big detail, but just to tell you that there's unique challenges. There's a challenge of low contaminants, low number of contaminants, challenges of the matrix that we're actually working with. And even when the bacteria are there, they might be too stressed to be recovered very quickly. And this goes on for routine food safety. But however, if we want to look at another issue, which is also a very important issue, even though it's a low frequency issue, we have to also think about how do we protect the citizens from potential bioterrorist attack. And in trying to do that, should there be an attack or a suspicion of an attack, there are challenges trying to detect the organisms that may be responsible. Yet, this is an important issue to deal with. So in all, I would like to think that we can get to a point where microbial testing in contaminating food or trying to detect contamination in food becomes a routine quality assurance process integrated to other systems in place where testing can be done at the site of production without interfering with trade. Trade continues, but testing is done such that before the food is consumed, the results are available and the chain is dropped. The chain is stopped there such that humans do not eat contaminated food. So what you're seeing here is a pictorial where in Canada, we have long winters. Our food, usually most of our fresh produce come from the United States, and either flown or transported by road, and it takes a few days. And the testing can be done before shipping, and shipping goes ahead. And if there's an issue of contamination, then the food product is not shelved and therefore protecting from illnesses. So this would be a proactive approach to a preventative approach rather than a reactive approach to ensuring food safety. So, and this is what we thought of a few years back when I had discussion with a gentleman to the left that you see on your screen there, Luke Masson, who is very busy with COVID now in Montreal. And he had just acquired one of the first two ion torrent bought in Canada. This was back in 2011. And we started talking about this. And this eventually culminated in Andrea Duprat, who works with us at the Canadian Food Inspection Agency here, going over to learn how to use an ion torrent. To cut a long story short, after many, many years and working at it very diligently, we eventually were able to develop this protocol, a culture-independent, genomics-based detection of Salmonella in lettuce, and this was published just a year ago. So what we like to do then is, can we now foresee a way of, one, turning this to a universal test, and two, being able to use it anywhere and everywhere to detect any or all microbial contaminants that might be in food. One of the things that we did in that paper was to evaluate different approaches, different methods of detecting Salmonella sequences from lettuce. And I won't spend too much time on this. All I want to show here is we eventually decided that the Cosmos ID platform was our best bet because it performed excellently well and it provided more information than any of the approaches that we used. And that's what brings us to the talk today. Now, culture-independent tests are now being recognized. They have there's some concerns. As a microbiologist, we like to see isolates, and I think that can still continue. But we can use culture-independent methodologies in a way that's fast and rapid, and as you would see, comprehensive and portable in a way that we will not be able to use any other method. So this is what we now decided to do with funding from the Canadian Security Science Program. We wanted to replace the ion torrent display with the mean ion sequencer using the Cosmos ID technology for our approach at bioinformatic analysis. And we felt that there are other tools that will add to the picture to make it easy to use this approach on the field. Now, with the funding from the CSSP, a group of us, you'd see the Army of Workers in a minute, embarked on this approach of first working on sample preparation, making sure that we have in our sample the target that can be sequenced and that's not overwhelmed by DNA from extraneous sources. And that took us many years. In fact, that took many years until we got it right. And then we went on to evaluate what it would take to be able to detect low numbers of contaminants. And we decided that genome amplification was a key aspect of the approach. So I'll come to that in a minute. We did whole genome sequencing using MinION, but we also used other platforms. And we used Cosmos ID for analysis, but at the same time, set up a database for in-house use. And finally, we did a field testing. So I'll go quickly through some of the steps. And here you see the army of collaborators that were involved, people very experienced and wonderful people to work with. And I'm so happy that our results bore the fruit of all the hard work that was involved in the various steps that we took. So what I'm going to do now is now go and start showing some of the results but before I do that, the culture-independent test that we developed is supposed to detect bacteria, viruses, or parasites. The universality is important. Remember, we have as many as 60% of organisms that are not tested for, but yet are causes of foodborne illnesses. So to do that, this is a simplified scheme of what we do. And I won't go through all the details, I'll just showcase the key point. The key point is that we start from the food sample, and in this case, we're spiking the food sample as a way to develop our test with known numbers of bacteria or viruses or parasites, and then prepare the sample and then spin our sample so that there's a pellet and there's a supernatant. The supernatant is used for viral detection and the pellet is used for bacteria and parasite detection. And then they now come together, we bring them together and then carry out a genome amplification and then sequence, will produce a DNA library and then carry out the sequencing on the MinION sequencer. So the genome amplification step, I want to highlight the fact how important this is. For anyone who's done sequencing, you do know that the amount of DNA and the quality of the DNA is so important. And so we felt this was a key part. Now in food microbiology, the pillar of isolating an organism is making sure you have enough of that organism through an enrichment process and making sure you have less of the undesirable organisms so that you don't overwhelm the system. In our case, the idea that we pursue was to amplify the DNA from everything that was extracted from our lettuce. And as you can see, amplification remarkably increases the amount of your DNA. And this is a key part. And so once you have the DNA amplified and we create a library, then we do the MinION sequencing. And I don't know how many people are familiar with MinION sequencing, but basically it is different from all the other platforms that we have, apart from the fact that it's so small. It's only weighs as many as 3 mice. If you have 3 adult mice on your hands, that's about the size of your MinION sequencer. It's portable. And it walks through the idea of a current passing through a membrane. And that current, if it gets disrupted because you have your DNA going through that same pore, through that same membrane, you create a signal that is then translated into bases, into nucleotides. And this will be a familiar one for those who've done this. So it's good to start sequencing and see a lot of green, things are working well. And if you can open the hood of the vehicle, as it were, and see a lot of neat things about amino sequencing. You can see the quality metrics of your approach. So basically, what you see to the left here is each of the 512 pores that are in the membrane, and the sequencing in time. So on the x-axis, the hours into the run, and on the y-axis is your read length. And as you can see, most of the pores here were working hard. A few pores were not sequencing, but this is a very good sequencing run. And then all the other metrics about, the quality of your read, the numbers that passed, and those are always fun things to look at. So once you have your sequence data, then the next thing is the analysis. And so this is where, and I won't be spending too much time on this. You already heard from Manoj. So we have an account, and we just upload our FASTQ data. Many years ago, when mineral sequencing started, you had FASTQ files that you had to convert to FASTQ and maybe to FASTStar before you do any analysis. But now things have progressed well. And those raw reads you can load directly into the Cosmos ID site. And you upload your sequence. The size of the sequences may be the longest, the most time-consuming effort here. Otherwise, once your sequence is uploaded, your result starts coming out very quickly within minutes. We have had sequence results show up within 2 minutes. Sometimes if the server is slow, it might take a bit longer. And this is an example of a run that we had. And what you see here is a bacteria sample, bacteria identified from the surface of lettuce. This sample was not spiked with any organism. I just wanted to show the list of the organisms. And these are common organisms that we see on lettuce. Sorry, it's close to lunchtime. I might be messing up your lunch here, but this is what we find on lettuce commonly. And yes, so let me now take some time to go through the data. And I think it's important to do this so that you actually see the results. It's a very busy slide. And so what we have here is a series of experiments where we chose 2 different viral agents, influenza A and hepatitis A, spiked lettuce with different numbers of platforming units, as you see on the slide, and run the sequencing. We took it through the protocol and then did the sequencing, and then the number of reads are shown. This is important for us to know how many reads do we need to be able to get a signal. As you can see, with influenza A, with about 500 platforming units in our 1 ml aliquot of the sample, we were able to pick it up consistently. Same thing with hepatitis A, at about 500 platforming units, we were consistently able to pick it up. This translated to 10 times the amount that was used to spike 25 grams of lettuce. So I just want to make that clear. So this is 500 plaque forming units detected in our 1 mil sample that we eventually processed. Now to salmonella. So again, the same thing. We get lettuce, we spike it with numbers of bacteria that we also count on an agar plate that we know what we're spiking. And as you can see again, we basically almost always detect the sample that's contaminated. We found that when it comes to the low numbers, having an incubation overnight before testing increases chances of detection, and as you can imagine, the bacteria multiplies more. So in one case, when we did a 3.5-hour incubation, we did not pick it up at six colony forming units. But otherwise, it works consistently well. And then we work on parasites. And these are parasites that were provided to us by Dr. Brett Dixon and Harriet Marks, and work together to carry out the work. And again, as you can see, at low numbers, very low numbers, 1 oocyst, 1 cyst, we don't see it, we don't pick it up. But from 5 and upwards, we consistently pick up, from 10 colonies and upwards, we consistently pick up Cryptosporidium parvum in this case. So then we now carried out experiments where we mix everything together, at least 2 or 3 agents together, and then take the product, apply the protocol to the sample of the contaminated lettuce. And there again, we use fairly high numbers here, but the same story, we're able to pick up the contaminants. Even more, when we have multiple contaminants, we consistently pick it up. So I wanted to make this case that we detected parasites and bacteria and viruses. Should there be a situation where we need to do that in our agency, we will have had to split up the sample and send them to different places. So I mean, otherwise, and we'll have had to send the virus, maybe to St. Hyacinth, which is about 3 hours away. We'll have had to send the bacteria maybe to Toronto, which is about 4 hours away. We'll have had to send the parasites to Saskatoon, which is 2,000 kilometers away. So while it is still important for us to appreciate the need for reference labs, for labs that are specialized in specific areas, you need that kind of expertise and that level of rigor. The need for us to be able to keep our consumers safe might require the ability to be able to detect all the parasites present in the sample at a location. So, and even if you have to do confirmatory tests later on, but this is a sort of thing that we think this test will achieve. Now, I want to take a few minutes to go through some of the details. So what I've done so far is to show us the development of the test. We were able to identify spiked organisms. We were doing the spiking, sometimes they give us the organism. We know what we were putting on. So it's a little biased. We know what we're putting on, but that was what you need to develop a test. Now, in this particular case, it's the same story, but it was a story, it had a twist to it, because we spiked lettuce with an organism that we have in the lab. And this is an organism that we've worked on we've sequenced it, we've done a lot of work on it. We worked on it because it has two related strains. In fact, two related isolates. To all intents and purposes, all three might be have the same strain. We found that if you are familiar with Salmonella, we talk about single nucleotide changes. And in these three organisms, they differ by a total of 9 SNPs. Through rigorous analysis, checking, mapping the genomes on top of one another, not using an algorithm, mapping the genomes on top of one another, they will differ by 9 SNPs. So to all intents and purposes, these 3 isolates are very, very related. They were all, again, obtained from the same location on the same day. Now, we just thought we'll spike the organism and use the organism to spike alleles and test. We did not know that Cosmos ID had that organism in the database. And so we matched that organism. We had sequenced it, we had put it in the gene bank. So it was a surprise that we matched the same strain and not the 2 other related strains. And then we've done the same thing again with the 2 other related strains, and each time they match the specific isolate and not one that's different by 3 SNPs or 9 SNPs. And so this was a very powerful experience for us to realize that we can do strain identification as long as that strain is in the database. If it's not, then it's matching the next closest relative to the strain. And that's the same picture I'm showing here, because Cosmos ID will present the results either as a table or as a bubble plot like this. And this is your Salmonella enteritidis in the spiked sample, but not in the unspiked sample. So then, how well is our assay working? We put things in there because we wanted to develop the test and we knew what we were putting inside. How about if we now ask other people to provide us with blind samples? And that's exactly what we did. This first one was provided to us in a tube. It was not on a lead tube. We didn't use it to spike lettuce. And the idea was just to test the system, more so the sequencing and the bioinformatic analysis. And indeed, we picked up all 4 parasites. They were put into the mixture. We did not know what was in there, and it was picked up to the species, Cryptosporidium parvum and Cryptosporidium hominis, 2 isolates that are not differentiable by all that means except some rigorous PCR. We were able to pick them up and differentiate them, and in the ratio that they were in the mix as we got to know. So that was very, very supportive when we got this result about 2 years ago. Now, then we ask one of our collaborators, Dr. Chenhua, to spike samples and to give it to us blind. And Dr. Sohail Noshad ran this test, and basically we got 4 different lettuce samples. We don't know what they were, and we tested them, and we ran them through Cosmos ID, and we hit all of them correctly. So that was, we felt that was a huge confidence booster for this approach. And then we now went on to test on recall food. These are food that have been recalled by our regulatory system using lab tests that are approved official tests. So we knew they were contaminated. We knew they were actually contaminated with Salmonella because we asked for food that were contaminated with Salmonella. And we're able to pick all this up. And in all cases, except one where we had the silver information, in all cases, we also were able to pick up a silver designation, which was not available to at the time of the recall. So this provides a power of the analysis that we're doing, that not only are we detecting organisms, we are identifying those organisms. And the other information which I will not be showing today is that we're also able to characterize these pathogens either through approaches like what phages are present. In case of Salmonella, that's an important one, or antimicrobial resistance factors or virulence factors. So finally, I'd like to finish with this. One of the things we wanted to do was to show that this test could be used on the field. So we set up a store. And this is the name of our store. And we actually go there anytime we're less busy. We hang around there a little bit, have a little bit of fun. And we decided to now test samples on the field in this store. And so again, we had blinded samples given to us. And all the testing were done in this location. And these are the samples that were given to us by Hongshen. He had spiked the samples, and in two cases, he actually mixed two different organisms. Well, one of them had two different organisms. Another one had three different organisms, but they had two of those organisms had the same strain, the same species. And in each case, we picked up all of them. Negative samples scored negative, positive samples scored positive. They were identified to the species, and we were very happy about this. So I would like to then conclude that by hoping that I've showed you our approach at developing a universal testing protocol to detect contaminants in food. We've had to work rigorously on sample preparation with all our collaborators who you've seen their faces involved in different stages of the process. We were able to define our detection limit, and we're able to perform the assay on both blind samples and record food. Finally, we were able to perform the assay in a simulated field condition. And with that, I'd like to acknowledge that the funding for this work was provided by the Canadian Science Security Program through the Defense Research and Development Canada. We're very grateful. In fact, the funding helped us to finish the preliminary work and to get onto this work, and we were able to do them very well, I think. I want to acknowledge Dr. Marco DiFuscio for his support, as well as project champion, Dr. Nancy Roth, who used to be the executive director of our lab here. Ray Theoret, my director, has been very supportive, as well as colleagues at the Research Partnership Management Division. Their support has just been very tremendous. And Tampola, Simone, Andrea, and Nish. Chief Science Operating Officer Proma Silva has been very, very supportive of this effort. Acknowledge his effort here. We are carrying out collaboration, validation of this test now with a sister lab in Burnaby with Katie Eloranta and her group, and that has been funneled through the CFI Food Research. Muel Mufti was provided some input early on. I want to acknowledge his effort. And we've had a lot of collaborators. Again, like I said, this is a work that's involved a lot of people. It's been done rigorously, and I think it's been done well. And with that, thank you for your attention.
Thank you so much. So we've got a bunch of questions. Maybe we can go through those one by one.
Okay.
And if you have more questions, please just post them in the chat, and we'll try to get through as many of them as we can. There's actually more than one question on whole genome amplification. Could you talk about that protocol a little bit more?
Yes. And if you want to have more details about that protocol, you can actually look at it from the paper in Frontiers in Microbiology. If you use my last name, you'll probably be able to pick it up easily from Google. What we used is a kit called WGA4, marketed by Sigma, and that kit leads to amplification of small fragments of about 500 base pairs on the average. Sometimes they are longer. You can get 2 kb, 3 kb. But the reason why we chose that was because it was the fastest protocol. We could complete it in about 5 hours. It was also one of the stronger ones. We had compared Sigma with Qiagen Repli-G, and Repli-G provide long amplification, 20 kb or even more. But the protocol that we had at the time was longer. Since then, we have now, with the help of the work of Tianbitan in the lab, we have now been able to modify the Qiagen protocols, and because there is some advantage in using log sequencing rate. So we will be evaluating that alongside the WGA4 amplification that we have. So that in a nutshell is the genome amplification procedure. It takes about 4 hours. We don't need to do DNA extraction for the bacteria. However, for the virus, of course, we were working with the RNA virus, which we had to transcribe to cDNA, and then we had to do the genome amplification. But all the genome amplification was done at the same time.
Thank you. Another question is, what was the total analysis time and the associated consumable costs per sample? There's actually 2 questions on cost.
Right. So we reckon that we can complete this protocol in 14 to 16 hours. We're trying to cut it down. But 16 hours, we'll be able to complete this protocol with the whole genome amplification step, full blast. One of the steps we can reduce is genome amplification. The other thing that we have done to estimate that 16 hours is we usually sequence our genome for many hours. We sequence the sequencing procedure is done for many hours. And we reckon that within 4 or 5 hours, we were able to detect applicants, whatever our target most of the time. However, there might be a way to rev that up because if you have a lot of targets going in, within minutes, you can actually pick up your target. Also, we're trying to use adaptive sequencing as a way of cutting things short and quicker, such that once we pick up a target, then the run is done. But this is where we know what we're looking for. Again, this is a universal protocol where you want to be able to use it to detect any and all contaminants. So in that case, you may have to go much longer, even beyond 16 hours.
Excellent.
Now, in terms of the cost, that's a good point. Right now, what we've done is we've been able to sequence 6 metagenomes at the same time. The big cost is the flow cell, which costs $900 US, except if you buy bulk and then the prices come down a little. And that is the big cost. The library preparation is about $100 for, I believe, 12 samples. So the more metagenomes we can sequence, the better. So if we can sequence 10 metagenomes, which are think it's doable. We haven't done that. We've done 6. You will bring the cost down to just a little over $100. But right now, it's closer to 200.
Perfect. I guess in terms of time, are you targeting 8 hours? Are you targeting 6 hours? Are you targeting 12 hours?
Well, what we're targeting is a good result, and whatever it takes, that's what it takes. If we are trying to detect any and all pathogens present, and therefore, if you have to take two schedules, 2 tons of work schedule to complete that, you will be much better off than you have to split your samples, send it to different places, and test for different things, and still wondering whether you pick up what's there or not, which takes a long time. So, for instance, we're going to culture salmonella by isolation. That could take us uh, 10 days. Uh, and at the end of the 10 days, you're still not sure what serovirus you have. In 16 hours, you can get to know the serovirus. In that case, probably sooner, 12 hours. So I think in time, it's possible to keep on refining the test and reducing the amount of time. But for now, what was important is to have a test that works well. And but that goal of reducing the time, it's not in our focus yet at this point, but we may look at that as time goes on.
Got it. The questions are pouring in, so we're going to go rapid fire now. How does a nanopore error rate accounted for in the pipeline, and what do you think the differences would be if you used Illumina or PacBio?
The error rate has not been a problem, and as you know, Oxford Nanopore it's just a different kind of a company. Their accuracy has been improving. I just showed you 3 isolates where they vary by 9 SNPs total, and we're able to pick up, to differentiate among those 3 isolates, so to speak. So the error rate has not been a problem at all, bar none. And certainly for the identification to the species level that we've had. Now, Illumina takes with a 600 cycle V3 kit, you're looking at 52 hours of sequencing run compared to 16 hours. Well, actually, compared to 3 to 6 hours of sequencing in this case. And your results will be available fairly quickly. PacBio, we have used PacBio, that was many years ago, and I'm not sure how their new platform is, if you can do metagenome sequencing or how well you can do metagenome sequencing. So I won't be able to speak to that. But with Illumina, you have an advantage of speed here with nanopore sequencing, flexibility. One of the things we have with Illumina is that if you run into a problem with your run and your machine stops in 1 hour, you're done. $4,000 gone. With nanopore sequencing, you can just add and start again. You can stop in the middle and start again. So the flexibility is amazing, and you have portability as an advantage as well.
Perfect. This one's an interesting one. So once we start using genome sequencing as the main tool to find pathogens, we'll also start to find other pathogens that we didn't expect in the sample. Maybe we'll realize that most samples may contain disease-causing species at a very low cell number. How are you going to define the threshold to categorize safe or risky samples?
Yeah, that's a good point. That's a very good point. And it's a very challenging question to actually address. for a number of reasons. When you look at it from a very academic point of view, an organism that's similarly non-pathogenic could carry virulence genes that could turn into a pathogen. So an organism that everybody says is not pathogenic could suddenly acquire properties that could turn into a pathogen. And there's no way of knowing that. And because organisms do live in communities, it's almost impossible to say very clearly that this will not cause disease, because that is a very definitive question that biology may not address that easily. However, I think from a regulatory perspective and from a practical perspective, then you can go from a virulence gene analysis, and this is what genomics also helps to do. If you understand the type of genes that could be present in that organism that cause disease, you could assess your risk based on the presence of virulence genes. After all, organisms acquire ability to cause disease because they have the property that makes them to inflict harm, and that's what we call virulence. And if you have a way of assessing that, and practically the only way we can do that is genomics. While this is a very difficult question to answer, it's also a question that people point you to the power of genomics as being able to resolve issues that we normally will even not want to touch because we don't have the tools. But with genomics now, you can touch that and begin to answer that question. Sorry, I don't know if I address it. That's the best I got.
Sounds good. What do you anticipate the day-to-day use of this method to be? And do you think government agencies are more accepting of it, or is it more of a still in the R&D phase?
We have not validated this test yet. Once it's validated, which we hope it will be, we hope to publish in the Compendium of Analytical Methods. And again, that is a discussion we're having with Sam Buhaja, Kitty Eluranta, and other people that are so much into this area more than I am. Once we have that published, they become a test that becomes an official test. An official test can be used as necessary. Of course, there's cost perspective, there's all that factors. The one thing that we can say, and I showed the slide, where the public health agency published on culture independent tests, the acceptability of this test is increasing. And so tests are developed to fit a particular need. And so if this test fits that particular need, I envisage it to be used.
Great. I feel the same way too. So I'm looking forward to your validation. This one's also interesting. So I think we'll have time for maybe this one and maybe one more question. How large a sample can be tested at a time? 25-gram samples are not very large, considering pathogen contamination is often not homogeneously distributed.
Yes, any size of sample can be tested. Do know that multiples of 25 grams are used in a lot of cases, including meat samples. So the size is not a problem. Any size can be tested. I mean, within reason, of course. But in the lab, people will use 25 grams, 75 grams, or I think 210 grams. There's that multiple of 25 grams that people use. So yeah, 375 grams. That's right. There's a protocol for 375 grams. So that is not a problem at all.
Perfect. Have you done any host reduction? Eukaryotic reduction?
I'm sorry, I missed that.
Have you done any host reduction to reduce the host so you enrich the microbial content?
We have not. Well, okay. The rigorous sample preparation procedures that we undertook, it took us years to figure it out, was really host reduction in that sense, because we reckoned that it was important not to have lettuce DNA get into the sample. Now, eventually it does get in a little, but if you get too much of it, you just overwhelm the system. So in that sense, we have done lettuce, we took careful effort to reduce the source plant DNA in our system. We did things like calibrating the stomach instead to make sure that the lettuce doesn't break up. Then we were we did filtration to make sure that chunks of lettuce did not get into the DNA prep step. So we've done that, and I think that's always going to be an important step to carry out rigorously for any new matrix that's been evaluated. Some may be easier to do than others, but that was what we did for lettuce, and that's what we hope to do with a few other matrices that we're hoping to work on in a little while.
Perfect. All right, last question. I know we're over by a couple minutes. Is it possible to identify which organism the pathogenic gene is found on? Or is it only the gene is present in the mix of organisms?
That's a good question, a very good question. If we do a long read, if you have a long read analysis, the results actually were small fragments. But if you have a long read analysis, it is possible, because then you can go back and harvest your DNA, harvest the sequences, and then look if it's big enough, you'll be able to identify the species and presence of virulence genes. So you're looking for virulence genes, and you got, let's say you got 50 kb, and your virulence genes, maybe they're just about 1 kb or 2 kb long. The rest of the 50 kb should be enough for you to be able to determine the species of the organism. Certainly, if you do enough reiteration, many of the fragments, you should be able to pick that up. So I'm optimistic that can be done. We have not done that.
Excellent. Perfect. Zeli, thank you so much for your time. I'm seeing a lot of messages saying amazing presentation, clapping emojis. I'm also getting texts saying amazing presentation. So again, thanks for taking the time, and look forward to speaking to you again soon. For anyone that we didn't answer the questions for, we'll go back through and follow up and make sure we get answers to you. And with that, this will be shared on our website, likely by tomorrow. So if you missed part of it, it will be available. So thanks everyone for attending, and have a wonderful rest of your day.
Well, thank you, Manoj. Thanks to Cosmos ID, and thanks to the audience.
Excellent. Thank you, Deli. Bye, guys.